Hello. Yeah, so I have to speak in English because I cannot speak Japanese. Sorry about that. So my name is Gojun Shen. So I work in IRCMS, International Research Center for Medical Sciences, which is across the street. And we work on a kind of cell biological problem called epithelial to mesenchymal transition, and shortened for EMT. So we try to study this epithelial to mesenchymal transition context in a few biological systems. I will explain a little bit of what kind of system we use to study. We study this epithelial to mesenchymal transition in the context of gastrulation, in the context of mesoderm formation, and in the context of cancer. So the reason is that actually when animal is developing, the cells organize themselves in a very dynamic way. And the most important aspects of this dynamic organization is called epithelial to mesenchymal transition here. So cells are either organized like a wall here. So we have a wall, is one side of the wall and the other side of the wall. So cells are organizing themselves in the same way, epithelial structure. And when they have to move around, they move around through a mesenchymal state. So in the adult case, in our body, we also are compartmentalized with lots of tissues. For example, skin is epithelium, mammary gland is epithelium, kidney structure is epithelium, the gastrointestinal organs is epithelial. But something goes wrong and actually causes cancer. So defects in epithelial to mesenchymal transition actually causes cancer in the adult state. So gastrulation is a time when you transit from a so-called pluripotent state, when this cell can become any cell in your body. And then they have to make a decision. And the first decision they make is called gastrulation. And they will become either called ectoderm, mesoderm, and endoderm. Ectoderm is going to become the skin and the central nervous system cells. And mesoderm is going to become your bone, your blood, your vessel cells, etc. And endoderm is going to become your gastrointestinal organs. So when the cells are making this decision, they have to undergo epithelial to mesenchymal transition as well. And the third thing that we are studying is so-called mesoderm. So this is the chicken embryo, and the embryo is here. We're studying so-called extra embryonic structure, which is the tissue which is important for the embryonic development, but after birth, they are no longer used for the storage. So in this three aspects, EMT, gastrulation, mesoderm formation, is the focus of our lab, so-called developmental morphogenesis. So morphogenesis means things move around, change shape. So we are interested in, during development, how cells change their shape, change their organization, how this change of shape and organization is important. EMT stands for epithelial to mesenchymal transition, and it's very important for development and for cancer. And cells are organized themselves either at epithelial structure or mesenchymal structure, or various shades of epithelium or mesenchymal. This transition is called epithelial to mesenchymal transition or mesenchymal to epithelial transition. And this is important in development as well as in cancer. The reason that I'm stressing here is that the cancer, most of the cancer is actually, the origin of these cells is normally organized as an epithelial structure. So if the cell cycle regulation goes wrong, you have a lot of cells. So this is called primary tumor. And primary tumor are normally not deadly. It's okay. Nobody died from primary tumor. But when the tumor cell starts to move around, metastasize, so-called, and that's when the primary tumor goes bad. It becomes cancer. And a lot of people, lots of money are involved in studying this process of metastasis, how they move into the blood vessel, how they move outside the blood vessel, and how they find a metastatic site and proliferate it again. So we actually collaborate with lots of cancer biologists to see what we can study, the regulation of this migration here during the primary cancer and the initiation of metastasis. So this is actually quite important. And gastrulation, as I said, is the so-called epigenetic landscape. Cells differentiate from pluripotent cell population to initiation of the 3 germ layers here. And in human, so these are the schematic, this is the real human embryo, this is the uterus, this is the implanted human embryo. And that's the beginning of the gastrulation. There's a structure called the epiblast. And then you have so-called extra embryonic tissues involved. So we are trying to understand the context of these cells when they eventually will become all the cell types in your body, as well as the context of this embryo in the face of the maternal tissues, how they interact with the maternal tissues, what are the functions of these so-called extra embryonic tissues, and how this little cell, pluripotent cell population eventually will become all the cells in your body. And so this is the kind of differentiation from epiblast to 3 germ layers, ectoderm, mesoderm, endoderm here, right? And in the context of the chicken embryo, the same thing. You have the extra embryonic tissues, you have intra-embryonic tissues, you have the intra-embryonic tissues, you have extra embryonic tissues. We're trying to study how EMT is involved in the extra embryonic tissues. So as you can see, we actually, it's a developmental biology lab, but we collaborate with cancer biologists, and also because I cannot speak Japanese, so people interact in English environment. So if you're interested in interacting with the other scientists in English, please come to my lab. Thanks a lot.